So let me start. Okay, so actually today I decided that I will tell you about a problem I worked like maybe 6 years ago when I was a graduate student at the Children's Hospital. Because this problem is the one that actually inspired what I'm doing now. So I hope my idea is that you can get ideas for future projects. So this is about the object recognition problem. So the idea is try to understand how the human brain works when looking at things. Okay, so here, as you can see, there's a person with electrodes in the head, and we are showing him a car, and from each electrode we get a signal, and the idea is to see if just by looking at the signals, if we can identify what this person is looking at. These are results published here in this journal and this paper, timing. So in case you want to look at it later. Okay, so now the idea is, okay, we show this person a car, so for each electrode we will have a signal. But actually we show different pictures, actually many pictures from different cars, from different shoes, from fruits, and the idea is to differentiate or to see from the signals which picture or what kind of object this person is looking at. Okay. So, okay, suppose that we have just cars and just shoes. They are all different, but the idea is if we can classify these two categories. And okay, okay. So in terms of math, basically we will say if it belongs to one category, we will give a number -1. If it belongs to the other one, we'll give the level 1. And then the different features that we can consider the signals are these vectors x. Although here everything is writing if we have just 2 features. So the idea is if instead of having the cars, the images, which we don't have, we have the signals that represent each of these cars. Okay. And using the signals, we would like to find this line, okay, this hyperplane that differentiates the 2 categories. Okay, so the idea is if you give me a new car, then I will check in which side of this line this object is. So if it's on the right, it will be a car. If it's on the left, it will be a shoe. Okay. So, okay, so the whole machine learning process is, okay, we have a lot of data, but we need to capture some features. Okay, we will see that in the MRI images we can use several features. We could just look at the texture of the image, although it might not any kind of feature. Okay, and then using those features, the idea is, okay, are those features enough to classify, to separate 2 different classes? And then to see if the classifier is really doing the job, we have validation data, which means, okay, I will give the classifier one new point and see we really know if that point to which category that point belongs. So we can check if the classifier is giving us is putting this new data in the right category. If it doesn't, after giving several validation points, we can have an idea how good this classifier is. If it's not good enough, then we go back to the training data, look at different feature extractions. Also, here I'm saying linear classifier, but there are different classifications. So this is a whole process where we can basically play around with data and with math models to see if we can get a way of classifying. Okay, so so let me give you a few more details about this experiment. There were 5 categories: animals, chairs, faces, fruits, and all vehicles, cars. And then, so the idea is, okay, so we have these 5 categories, but we show one patient, a lot of images, okay? A lot of images from the first category, a lot of images from the second category, and so on. And then we have a lot of data, right? For each image we are showing, we will have, for each channel, for each electron, we will have a signal. So the question is, okay, that's a lot of data. So we need to reduce the data, get some feature extraction for all that data. So here, for example, you have a signal. In this signal was all processed. So there was some convolution, and then this will smooth and get rid of some noise. Then some Fourier transform, and then you have a lot of frequencies now, and then the frequencies were put in different groups. So here, for example, there are 5 groups. So you can think of, you have this signal, and suddenly you have only 5 numbers related to this signal. So this signal was basically replaced by 5 numbers. Okay, so there was, we can say that in that sense, there was a reduction of dimensionality there. And something, keeping with the whole process, for instance, here, I just said that for each signal, now we have 5 numbers, but maybe for each channel, only 2 numbers were more relevant. Okay. I don't even remember all the details, how we get to only these numbers, but my point is there is a whole processing of the data to go from, the high number of the high dimensional data to just have, let's say for this channel, 2 features. Okay, and okay, so we can run also the classifier, and so basically now I'm giving, so I have a classifier which I'm giving for each electrode, let's say 1 or 2 numbers, and I run the classifier, and the classifier also can tell me which electrode is more relevant. Okay. So here I have this on the left are different categories. We identify that for the first category, only these 2 electrodes were relevant. For the second category, these 3 electrodes were relevant and so on. So here you can see that we look at the performance of the classifiers. By the way, we have 5 categories, so the chance is 20%. Okay, so here you can see that the this classifier is classifying, but not just by chance. Okay, because these are higher, much higher numbers. Okay, so that was everything I wanted to say about that experiment. But then let's go back to Chiari, and the idea is now we want to differentiate, let's say, healthy patients from Chiari patients. Okay, so somehow we could just take the whole image and try to differentiate, but that's a lot of data. Okay. So the idea is, okay, let's try to get some features from this data to reduce our the dimensions to play with. Okay. So the idea, okay, so what is Chiari? And the idea is, okay, we need to have categories, and we could just have Chiari against healthy, or depending on which data we have, we could try to find a classifier or a way of separating these 4 groups, where here I'm thinking, okay, so here you have this set on the left is the tonsillar descent, people who have a tonsillar descent of greater than 5 millimeters. And this set here is formed by people with Chiari-like symptoms. Okay. So the intersection is what we really call Chiari patients. And then here the set of people with tonsillar descent greater than 5 millimeters, but without any symptoms, is the asymptomatic Chiari patients. Then we also have these sets of people with symptoms, but that don't have the tonsillar descent greater than 5. So we can really have here 3 sets that we can consider categories, and then we have around outside the healthy patients or healthy people. So it will be really nice, I guess, to have to be able to have a classifier or find a way where we can separate these 4 groups in a very automatic way. But okay, we have a long way to go. Okay. Okay. So our first idea was, okay, we have MRI images, but we need to have some feature extraction. We cannot just use the images. So we thought, okay, there are a lot of morphometric studies. Why don't we use some of these measurements? So there are several studies already. Secula, most of these papers were focused on trying to say why, or trying to justify that the brain is small. Sorry, that the cranial fossa is small, and this paper by Secula was trying to basically separate these patients, the Chiari 0 patient versus healthy. Today I will show you some results where we are trying to separate this group of tonsiliar descent greater than 5 versus healthy, and also to separate these two groups, the asymptomatic and the Chiari patients. Okay. So there is no other separation at the same time of all these groups, not yet. Okay, so the idea, we focus in Urbizu paper where they have 19 measurements. Okay, so okay, so here I have basically the this image is done by Daniel McQuaid during the last summer at the Chiari Research Center. And okay, so the data we use was we have 25 patients, 8 healthy, 9 symptomatic patients, and 8 asymptomatic. Okay, so and the way what we did is the following. Instead of using the 19 measurements at the same time as a classifier, it was too much for the number of data points we have. So what we did is we just took pairs of the 19 features and ran classifiers for each pair. Okay, and then measure the how good each classifier performed. And here we could see that some pairs were better to differentiate between Chiari patients and healthy people. Okay, and that's the ones that we show here. That's the nice thing about also using pairs. We can look at the data and plot the data here, and you can see here on the left you have Chiari patients, and then here on the right are points corresponding to the healthy people, and we plot the classifier. Okay, so basically this line is our classifier. Is telling us people on the right should be always healthy people, and people on the left should be Chiari patients. And this classifier is using the Wakeman, I'm not sure how to pronounce that correctly, Wakeman angle and the distance to the bones. And here there is another classifier where actually you could see that even though we are using a pair of features, you can see that the division is really, the separation is really given by the Wakeman angle. Okay, so actually now we are exploring some mathematical techniques so that we can kind of project and choose the features in a more automatic way. So we expect that if we use this reduction of this dimensionality, it will just give us the angle back and not the width of the PCR, because here it doesn't really differentiate, just the angle. Okay. And we did the same, but here we are only taking the asymptomatic versus Chiari. And we see that there is one particular classifier that got the best performance comparing with the rest. So we look at that. That was the classifier that uses only 2 features: the distance to the pons and the width of the foramen magnum, and so again, here we have two features, so we can really plot the data and the line to see this idea of classifier, where we have one category in one side and the other category on the other side. And again, you can see here that symptomatic is on the, let me see. Yes, symptomatics are here, and then asymptomatics are on the left side. So the Chiari patients are the ones that have a smaller distance to the pons. Yeah, something that Bryn was talking today. Okay. So let me kind of summarize a little bit. So basically, we found, you know, a way of classifying Chiari patients from healthy ones using two measurements, okay? And we got this classification rate, but remember also that we have very little, very limited data set. So, but this is what we have. So we are now looking at more data points and, again, playing around. And our idea is that trying to find a way where we can separate these categories by trying to let the computer do the job. Okay, we don't want to influence. We want just to give the data, play with mathematical models, play with different you know, way of writing the data, and see what happens. Okay, so I'll take questions. Questions?
Sure, go ahead.
So you showed us great talk, by the way. You showed us two histograms for.
Oh, I cannot hear you. Someone next to the computer will have to repeat the question. But
okay, can you hear me now?
Okay, let's try.
Okay, so on slide number 22 and 23, you showed us two different histogram, I believe. So what's the difference between these two? I mean, the
okay. Yes. So let's see. So in this one, you have Chiari patients and asymptomatic altogether versus healthy patients. And in the next one, here we only took the asymptomatic and Chiari patients. So we didn't use the data from healthy patients.
Okay, so for the first, can you go to, yeah. So what is the, on the vertical axis, what is the #1 classifier?
So is the herniation. So here it's saying, yeah, the herniation can classify. That we, yeah, but this is, I mean, we are kind of using the herniation to label the data.
So as I'm seeing here, the herniation is still a significant factor, right? Because it's very rare.
Yeah, but on the other side, it's also used to label.
So it's a little bit it's not very useful because the cutoff for the patient is 5 millimeters, and the healthy has to be less. So therefore, we expect that because the learning data is 100%.
This is kind of pretty important.
Yeah. So if we don't get that, then there's something wrong with what they're doing, right? But it is interesting that many other variables, because this is a linear addition to the classifiers, resulted in.
Malena, I have a question. I read your abstract as well, but would you be so kind and tell us in slide number 24 what exactly the Wackenheim angle is, and what the distance to the pons means.
Oh, okay. So here, maybe I will use Urbizu's data, that looks better. But the distance to the pons is this one here.
Sorry, can you make it bigger? Difficult.
Yes.
If you just
Maybe
make it bigger.
Here, can you? This one is good, or?
Yes, yes. So how do you define this?
So I can answer that a little bit
actually,
better.
I don't know.
Malena didn't do those measurements herself. She took the data from us, and basically what it is it's from the tip of the clivus to the what do you call it? Those two bones are the basion-opistheon line. Okay, so we draw that line from the basion to the opistheon, and then
can you say which letters are these? The abstract wasn't sufficiently clear to me to tell me what we're looking at, because it was using also letters for both angles and points. So forgive me for asking.
Yeah, no,
which points?
So if you look here,
which points are defined by defining the Wackenheim angle on these pictures here?
Right. There isn't any. Actually, that's ambiguous.
It's C and B. This is the same here as this, right?
Yeah, I can't hear.
This is actually from the Urbizu paper, and it's confusing on that, right? Because they don't. You're right. They have multiple letters for different parts of the inset, right? But the Wackenheim angle is going to be the angulation between basically the tip of the odontoid there. See that? Right there, and going down, and then along the slope of the clivus. That's the Wackenheim angle.
This is actually C right here, Brent. So I think it's from E to C, right?
That's for the pons, though.
Yeah,
he was asking about the Wackenheim angle.
But before
so the Wackenheim angle is the angle between the clivus and the axis of the vertebrae, yes?
Exactly, yeah. So it's not D, for example, going from the tip of the odontoid down to the central point of the C2, C1, C2. It's along along the vertebrae.
Okay, and the other one, the distance to pons, what did that mean?
The distance to the pons is just like I was saying. It's the so you take the basion-opistheon line, right? So basically the foramen magnum line, and measure the perpendicular distance to the upper aspect of the pons. So it's the tip of, you see E, the letter E?
Yes.
Going down to that line that goes across the foramen magnum, basically.
And that line is C. You can't see it right.
Yeah, it's kind of hard to see. So we actually have the problem is.
So it's the angle between A and the tip of the clivus and A.
What do you mean?
C.
In your figure A at the top left. Yes.
Not the angle.
Yes.
No, it's not the angle. It's a distance. So that's what you're confused by.
Which one? Which distance?
For the pons, the measurement of the pons is a vertical distance from the line, the basion-opistheon line, the vertical distance from there to the upper part of the pons.
So that is E to C,
yeah.
If you look carefully on that upper left figure.
Okay.
Figure A.
This is a common measurement. So, the basion-opistheon line, that's how you get the tonsillar herniation. That's your line B, right? And in many other papers like Sakula, many other papers in the literature, they take that line, the basion-opistheon line, and basically make multiple measurements perpendicular from that line, such as the tentorium, as you see, or to the fourth ventricle there, or to the tip of the tonsil, or to the upper aspect of the pons. Sometimes they do it to the lower aspect of the pons, but it's a very common measurement to make this.
And what's the justification for that line?
What do you mean by that? The justification,
well, you might as well measure anything.
That's true.
What is the reason for wanting to measure just that line as opposed to something else?
So in this paper, they measured 19 different measurements, right? So they didn't only do that one, but the justification specifically for the pons, and especially the upper aspect of the pons, is that it's very well delineated, right? You can identify it pretty decently on a mid-sagittal scan, and many of the measurements are like that. I mean, they need like anatomical landmarks that are somewhat easy to differentiate, you know? Obviously, you've probably seen some of the papers by Noel Malperin or different groups where they're doing automated techniques to try and get whole volumetric analysis. That sort of takes some of this out of it, but even still, there hasn't been very good conclusion that, for example, segmentation of the cerebellum is very consistent, or the brainstem, for example.
But Brent, wouldn't it make sense if you assume that since the main fiber tract pass up to the rest of the brain go through the pons, the reticular activating system? So that would be very pertinent, because if you had problems with those fiber tracts, you would expect that to be the most important.
Did you hear what Phil just said?
Yeah, kind of, but that's sort of an ex post effort of explanation, because we are if we took that line, that vertical line, in any way seriously, which I don't, developmentally speaking, this would be produced out of two different, completely separate processes. One has to do with the way of how you bend the pons, if you will, or how you grow the pons, if you will, and the other one the other component to that would be the length of the clivus and the angle of the clivus, both of which would work into that single parameter.
Yeah, so I mean, so in terms
and
of
that is a big problem, because there's no biological justification for using that line that I can see here.
I mean,
because we know that the clivus is even made of at least 2, if not 3, separate growth centers. So the length of the clivus is obviously something that's going to be important in some way, and some aspects of that length will also have an effect on the angle overall, of course.
This is Phil Allen. Let me take a stab at this. So let's say, just in terms of a fiber tract model, if you're thinking of say the reticular activating system being affected, and let's say shrinkage of that distance from the top of the pons down to that foramen magnum line. If shrinkage represents damage to those fiber tracts, wouldn't that make sense from a neuroscience perspective? That people that had more symptoms should actually show more fiber tract damage, as indicated by shrinkage of that line segment.
Well, this makes assumptions about the normal length of fiber tracts among normal people and symptomatic people that have never been verified, or have they?
Well, but why would you, is there any reason to assume that the fiber tract should be shorter for any other reason other than to atrophying? Now, what I agree, it would be nice to show someone.
Has anyone shown by tensor MRI or any other method that whether or not there is actually a change in the length of these tracts?
We're doing that right now, by the way. We're starting that study right now.
There are groups that
but nevertheless, you can obviously you could obviously, from a mechanical point of view, late in life, think about that as a potential parameter. However, you might as well take any other angle between this point E and some place at the clivus as your measurement, as your parameter. I don't see do we know mechanically that only the distal tip of the clivus would have a damaging effect as opposed to some other regions of the clivus?
We don't. Yeah, no, we don't really know. I mean, these
No.
are just measurements to start getting at those kind of things, right? I mean, this is the utility of doing the machine learning is you can take in many variables and see which
Yes.
ones are decent. And actually, in her talk here, she's just using specific measurements.
Sure.
We want to go to
I'm,
an automated.
I wanted to illustrate that with a little neuroscience example here. Malena, you mentioned the identification problem of cars and stuff. Now, the problem, obviously, with establishing a classifier is that your information needs to be granular enough that you have enough information in what you put into the system to get something out. And as you know, beautiful work by Christoph Koch and other people have shown that there is such thing like a Jennifer Aniston neuron in your brain, that the information about, say, even a single person or anything you like can be extremely fine and extremely granular. And in this case, if you were to make field measurements of the kind that you've shown us, you would never find it. And here we have kind of, I think, an equivalent to that situation, because what one really needs to look at is how these individual parameters, that have been put into models are constructed in themselves. Because the ideal would be if you could apply this machine learning to a far more unbiased way of establishing parameters than just taking the ones that were handpicked by some clinicians. Even if they're widely used, it's wonderful that you actually see something that can actually be a pointer towards something interesting. But I think it will be very helpful if one could cover this region of the skull with as many possible points for morphometrics as is reasonable, because I see, because the clivus grows as an independent entity of the brain above it, I see no biological justification for starting with that distance in the first place.
This is Phil Allen again. Just a question of curiosity. What if you said, all right, this is a data mining exercise. It's purely data mining. And then you find out what variables predict and then worry about the other stuff later on. Because what you're talking about in Koch's work, that would be using functional magnetic resonance imaging and seeing where objects are activated in the fusiform gyrus. This is a morphological study that is just looking at data mining. Wouldn't it be helpful to at least have predictors of whether people were Chiari or non-Chiari, and then worry about why it is later on?
Yes, that's wonderful, but sadly there isn't one around, as you know.
Yeah, but
and that's the biggest. This is the biggest problem so far.
But
that
that's what
the bona fide predictor that people choose in the past, like tonsil herniation, are totally useless, as the colleague in Michigan and other people have shown.
So actually, Malena, can you go to the next slide, like maybe 2 slides later, or keep going? Okay, one more. And then one more. Here you go, right here. So speaking of that, how there isn't really a very good predictor, tonsillar herniation doesn't work very well. I think that's the interesting thing about this, is that, using this technique, she identified that the measurements that separate the healthy group from the Chiari group, right here, Wackenheim and the Pons, were entirely different than the measurements that separated, for example, the symptomatic and asymptomatic group. This was just the foramen magnum and the Pons, right? And that did not include the herniation, actually, right? So the herniation in the symptomatic and asymptomatic group, those are not, that's not as good of a factor as the foramen magnum, the width of the foramen magnum in combination with the distance to the pons, right? So it's like we're finding out.
Bryn, can you define this again? What you mean? Which measurement that is? Foramen magnum? Do you mean the width of it, or?
Yeah, the width of the foramen magnum from the basion and pistion, basically from the tip of the basion to the tip of the pistion, and the distance to the pons is like we were talking about. So she has it labeled here. And the very interesting thing is that this, for example, another interesting thing here is that the Wackenheim angle, when you plot that and you look at it alone and you say, is the Wackenheim angle alone different in the Chiari versus the healthy group? They're not. There's no difference. But when you use this machine learning technique, when you take it into account in addition to the distance to the pons, suddenly it pops out and says when you use them together, it's meaningful. But when you use Wackenheim alone, so this is like the old techniques that have been used by the medical community. Let's look at the Wackenheim angle. Does that help separate the group? Is there a statistical difference? What we're saying here is there was not a statistical difference in the Wackenheim angle, but when you take it into account with the distance to the pons, using this technique where you didn't ever show the equation, Malena, where it's a coefficient multiplied by that first value, Wackenheim angle, plus another coefficient multiplied by the pons, and then that's equal to the classifier. I don't know if you had that equation, but I think it's extremely interesting that these different parameters are popping out that you normally wouldn't see. They're not tonsillar herniation for sure.
Yes.
Go ahead.
So I find this extremely interesting from a developmental point of view, because clearly the distance to the pons is, developmentally speaking, basically a direct outcome of what we would call dorsoventral patterning of the occipital region, of the exoccipitals and the supraoccipital. And so the cells that invade into this region and are responsible for the dorsoventral growth, which will directly affect the length, what you call the pons distance, these cells are also invading into the clivus and are also partly responsible for the overall length of the clivus, but only partly. So what I want to say is that there is a potential dependency of these parameters among each other, genetic one, because it's the same process of growth, and probably even the same cells that are affected either of the two parameters. And if you can pick out that this is an important criterion, that would be lovely.
So one thing to state is obviously the data that she showed. Sorry, Malena, I'm kind of speaking for you here, but it's extremely limited data set, right? We only had something like 10 subjects in each group, right? But now we do have all of the data from the Urbizu study. They actually gave us all of their 19 measurements in 100 Chiari patients that are symptomatic, 50 that are asymptomatic, and 50 controls. So we're going to test the same algorithm on them. And I'm wondering, Malena, did you get a chance to process any of that yet? Did you have any?
Yeah, we were playing around, and basically, actually, one good classifier was if we take the sum of these three measurements, kind of, and we were calling it the total descent. We have 80% accuracy to separate Chiari patient from healthy.
So
but this
the big thing here is that it looks like many structures are low, and it's even structures that are very far away from the clivus or the foramen magnum. Right?
Mm-hmm.
So go ahead. Phil has another.
Malena,
Yes.
how do you deal with CM0? Because that's not healthy, right? Those are people with no dissension but have the symptoms, right?
Right.
So, so you're really
yeah. So
go.
no, see that I don't think we have data to deal with that right now, because what we have is from Urbizu's data. The patients that have let me think.
No, I think you're actually not. I don't think that's correct, Malena. I think that what they gave us is they gave us obviously the Chiari patients that have more than 5 millimeter of descent. They gave us the healthy subjects, and now they've given us
mild.
the ones that are mild, the ones that are like Chiari 0. They do not have 5 millimeters, but they do have symptoms of Chiari.
Yes. So
So
yeah,
we
yes, you're
haven't looked
right.
at those, but
I predict they're going to be the toughest ones to deal with because they just are different.
I can give you an example. We have one subject in a current study that we're doing where the tonsillar herniation is actually -1. So they're a Chiari 0. And we looked at it and we thought, whoa, why are we classifying this as Chiari? Well, I looked at the 19 morphometrics on this subject, and the distance to the pons was reduced, actually is one of the most low, smallest distance to the pons of any subjects in the entire study. So I don't want to say like the distance of the pons is absolute, but it conveys like, this herniation, obviously it's important, but if you have some of these other measurements that are very small, then maybe that's more important. I don't know.
Is it a fair
Is it
question?
possible?
Is it a fair?
Is it possible?
Go ahead.
Is it possible to get, a really, really large data set like the data set that McCormick in Michigan had, so that you can really look at the Wackenheim angle and the distance to the ponds in, say, a few thousand people?
So
because I think really the quality of your classifiers will change so much if you really look at loss. And also look at different age groups.
Yeah, so in that data set, we actually had them speak last month for our group meeting, Jennifer Strail and Dr. McCormick. We actually do have a collaboration going with them to get their data, and we already have a whole set of pediatric data. But I want to point out that their Chiari subset, so they had 24,000, something like that, MRIs that they assessed. They randomized sort of, selected ages, different age groups from that, and then boiled it down to something like 1,200 MRIs. And then of them, only a small subset was Chiari. It was like, I want to say a few hundred. So
yes.
So in the end,
but it will be important to have that, as the baseline to really know what is the expected normal or symptomatic or asymptomatic situation.
But I'm just saying, actually, with the data we have right now, we already have a similar number. So the Urbizu data, like I said, is 100 of Chiari, 50 that are asymptomatic. So it's basically 150. And then from several other studies we have going, we easily have 250 or 300. That would put us at a number higher than the Chiari subjects in the Michigan data. And for the controls, we have a lot of data as well. So definitely we want to go in that direction and verify with, greater accuracy what is normal and what's not. So
could you consider that maybe that distance from the peak of the pons to the foramen magnum, if that decreases, and again, you could overcome those psychometric issues, could you assume that that's a measure of a different type of compression from the top rather than from the posterior to the anterior side? And this is a question for a neurosurgeon.
I don't know if any Cletus is on. I don't know if
but you see what I'm saying? Maybe that is a measure of compression, actually.
Cletus, are you there? Can you hear us?
No, he has to go.
Oh,
he left a chat message.
Okay. Well, he might be able to give us an idea. But All right. Well, does anyone else have
I
any?
have one more suggestion along the lines I mentioned. So if you look on this slide here, if it is possible at number C, the bottom left hand, if it is possible to look at the distance H to C, that is very important because that's going to be subdividing the clivus into two parts. And at the position where you see the H, this is the growth zone of the clivus. Yes. And sometimes even in humans, it doesn't even close properly. So that will be a very important point because I would expect that the distance between H and C is correlated to the distance between C and B.
So actually, that's a great idea. So one thing we could do is, this is a T1 weighted image. We could do like 3D reconstructions of just the clivus bone. You know?
Yes, but the important thing is that you subdivide the clivus into, you know, you can see the boundaries there. You see this whitish sheen, which
Yeah.
is where the H is. That's an important subdivision because the bit in front of the H, towards between H and D, is of a different origin than the region between H and C.
Yeah, that could be done. I mean, it's a little bit hard to differentiate, objectively, but I think we could, based on the intensities there, it's possible that could be, reconstructed in 3D and then split into two regions and quantified. Um, you know, yeah,
To see if you find relationships here
yeah.
that are good
Yeah.
predictors.
Maybe we'll get a student to work on that. That sounds interesting. Yeah, just the clivus bone assessment. So, yeah, maybe we'll send you an email of a reconstruction and you can check it out.
Mm-hmm.
So, anyone else have comments?
I've got a sort of more general comment.
Can you
So do you?
try and speak loudly?
Okay, I mean, I don't even know if this is, would be address this, but is there a good idea of how all these parameters are changing with each other? I mean, which ones are interrelated? Because you seem like you're measuring a lot of things that might be one parameter that's affecting all these other parameters simultaneously, trying to use statistical shape matching or other statistical approaches to figure out which of these parameters are interdependent on each other to then identify the one independent parameter that's kind of driving several of these relationships. Has any work on that been done?
You can look at the squared semi-partial correlation to look at the unique variance, but I doubt that's a completely linear. They're not doing regression per se, but traditionally you would look at the unique variance with the squared semi-partial correlation to rule out multilinear.
Yeah, I'm just has that ever been looked at? Have you ever tried?
If you're interested in helping us on looking at it, we have all the data to show you. Just punch through it and see if you see something different. Or
I would be very interested in that from the point of defining actually what you're going to measure. You might want to have a look. There's studies, for example, morphometric studies in mice by a guy called Chevrod and Atchley. And these people have been parameterizing mouse skulls with great precision, and they have not made any major assumptions as you're doing here on what should be correlated or what should not, or what should be responsible for a symptomatic phenotype or not. But if you do this with sufficient resolution, you'll find modularities within your data set, and that's obviously what you want, because what will happen is that there'll be many internal dependencies between parameters, and only some of which will have then a symptomatic outcome. But without seeing the whole picture, you can't actually make any ad hoc guesses here.
yeah. So I think we're a little bit over time here. I know we started a little bit late. I'm going to try and do better next time, not getting started late. Does anybody else have any last comments? I know Mark Quigley's online, and he didn't say anything. If he has any questions or comments, I think his microphone is muted. So, okay. Well, with that, we're going to end the meeting. Thanks everyone for attending. And next month, you saw the date on the schedule. Just keep that in mind. Have a great day.